The discussion. Brian Riddell. So Brian is, as I mentioned before, the president and CEO of the Pacific Salmon Foundation, and is a very well-known expert in the salmon world. We're just delighted to have him here today. He, as I mentioned before, he trained as a geneticist at McGill, and in fact we overlap back there, so we have to compare some notes. And has won many awards for his expertise in this area. And again, Brian, thank you very much for joining us, and look forward to you moderating the session.
Thanks, Brett. All right, lots of cheat notes for you. Thanks, Brett. I didn't know we overlapped. I don't even tell people that I was in genetics, to be honest, anymore. Because I'm not sure that we actually talk the same language anymore. And after listening to the last two talks, I know that we don't. I did have one little point of humor as you brought something into mind. The last meeting I went to at the Cohen Commission, there was a question posed from the Cohen lawyer, actually, that isn't it true that DFO has never done anything right in its history? I'm not sure they got through the question more as a comment, but I have to say that in the sort of work that Kristi has brought to salmon, we definitely did the right thing. We brought Kristi in. So it's been a joy to actually see the potential of these tools. And now I think with the BC Shellfish Center that you just heard from Helen, I think that's going to be a really important development in British Columbia. In a week, the Royal Society report on Canada's oceans and biodiversity is going to be released here in Vancouver, and that actually at the other SFU downtown center. And the whole issue of aquaculture will be brought forward in that report, including shellfish culture, because it's very important on the East Coast, growing on the West Coast, and as we just heard, it's got great potential, but challenges with climate change. And the thing that really struck me in looking at the effects of shellfish culture on the West Coast is we have limited areas that are really high quality for environmental fitness for the growth, and so it tends to be very intensive. And that in itself generates a public response that maybe by doing what we've just heard, you could develop strains that are more suitable for a more intensive culture and maybe reduce some of that spatial footprint, and it would be a more positive effect for British Columbia. I think the appropriate thing to do would be to start with some questions the way Stephen did, and I'm sure that both will generate some questions from the floor, so why don't we do that first?
Oh yeah. So I work with mammals, and in mammals all stress is not bad. In fact, a certain amount of stress is important. And so I'm wondering, in your system, is there any way that you can tell the difference between stress, which is, making the animal stronger because it's turning on different enzymes and stuff and can handle, movement and things like that, versus stress that is actually pathological?
Is that
so? Okay, Helen.
Okay. Well, at the moment, we're just looking through all the data that we have. I mean, there's ways that we can find through certain pathways, for example, genes that perhaps would be indicative of what is a reduced stress, but a positive stress, like in your example. But it's just early days yet. We're just going through the data. But I fully anticipate that there's going to be X amount of genes that we start with, and then we start whittling them out for the ones that we really think are potentially involved in detrimental stress responses, and then those would really become our, extreme end biomarkers for the future.
Can I just add quickly to that? I agree with you in that that is one of the challenges in the work that we're doing. Interestingly enough, fish that carry the MRS signature that we've defined in multiple tissues, we basically have done experiments where we're tracking the shift in physiology over migration, and we can look at the shift in physiology over migration at fish that are positive for that signature and negative with that signature, and we find that for instance, there is I think a fairly normal stress response that occurs when salmon move between freshwater and saltwater. And I think that that stress response is highly adaptive and very important in their ability to adapt to that new environment. Fish that carry this MRS signature have a much reduced stress response when they move between those environments, and a response to high water temperature stress as well. They're not responding in a typical way that we would expect would actually be now, or would be adaptive for those animals. So yeah, it is more complicated than simply looking for any old stress response, because many times they need those stress responses to survive.
Hello. Okay. Hi, my name is Helen from the Genome Sciences Center. I think this question is mostly for Kristi, discussing about the viruses, for example, parvovirus and the influenza virus. I'm sure there's efforts to be looking at where the root causes for these viruses to infect the salmon?
Yeah, so we're now carrying on challenge work, and we're looking at vertical and horizontal transmission for the parvovirus. And that's ongoing with UBC and with a virologist at DFO. In terms of the ISA, I mean, there's a lot of work that's already gone on in that in Norway. We don't really, it appears that what we're picking up is a novel strain of an ISA-like virus that has been here for at least 25 years, because we've been able to pick it up in samples from 1986 in our DNA archives. Or RNA archives. You know, we know that ISA is largely horizontally transmitted, but can be transmitted through eggs as well. That's not really totally my area, but we're doing similar sorts of studies with parvovirus to understand transmission dynamics, because that ultimately is very important if you're trying to make decisions on how one would mitigate any such thing.
Hi. I'm Robin Koo from the Genome Sciences Center. And as the engineer there, I make it a habit to ask one naive methods question at each one of these meetings. So I was wondering if you felt that it would be desirable to be able to detect known viral transcripts of lower abundance than you have, because you're sort of doing this, you're looking for these targets in these slippery fish. And do you feel that there may be viruses that might be there in low abundance that you can't detect with the methods you have, or do you think if you were able to detect low abundance, you just see all kinds of noise that wouldn't tell you anything?
Well, actually, we're doing a little bit about that. So the fluidine biomark system, I think we're one of the first labs to actually utilize that for pathogen screening. And the literature, I mean, it has been developed for qPCR analysis and other kinds of assays, but it is actually more sensitive than the 7900, which is a typical qPCR platform. And this was hotly debated when I actually testified for the Cohen inquiry, because I was using a different method of detection than other labs have been using, and I argued that it was a more sensitive one. It detects transcripts at a lower copy number. But the other thing that we're following up on as well is using high throughput sequencing. And so for this was successful so far with the MRS signature. We have other kinds of signatures that indicate differences in immune stimulation that we hypothesize could be pathogenic. So we're screening fish that carry those signatures with various pathogens to determine if they are associated with any known pathogens, but we're also conducting high throughput sequencing to see if there are other pathogens that we don't know about that may be present. But sensitivity is certainly an issue. It was an issue a little bit in a parvovirus challenge study that we did recently, because when we first identified positive and negative tissue fish, we used a traditional PCR test because we didn't have a quantitative test. When we went back to those same tissues and we had a quantitative test, we found a low level of positives in some of the fish that we had identified as negative previously. So sensitivity is always an issue. In terms of people who work on fish disease, they kind of threshold their sensitivity towards levels that are known to be associated with the disease state. And so they're quite happy with saying, you know, we're looking at sensitivities that indicate that there's a disease present, not merely a virus present. In our case, we really just wanted to know what viruses are there. We'll later on look at what diseases are there and what diseases might be caused by those. But yeah, sensitivity is an issue.
Thank you. And as a dedicated listener of radio and reader of newspapers, let me thank you for making this field so exciting.
I'm surprised that Kristi didn't use her Time magazine cover in one of her slides. It was well done and fooled me for a couple seconds. Any other questions? Right.
So I guess I have two questions. So maybe for Gordon first and Helen. I guess, it's interesting. Helen, when I came to visit in Vancouver Island University and visit the operation, it was interesting to kind of learn about the different shellfish that are harvested and the economic value of those shellfish. And so my recollection was that mussels are kind of at the lower end of the economic ladder, if you will. And as I recall, geoduck, I think, is at the other end. And so, I guess the question is more just a general one. As this technology evolves, I mean, it is farming, and you really made that point to me. And it really struck a chord that I guess in this farming environment, can industry actually, I guess I'm looking at the receptor at the back end, at industry being a receptor for this. And so Gordon, it's more to you, I guess, is that can you kind of, I mean, other than yield, is there an economic indicator that you're really most focused on here? Is it trying to prevent disease, whatever? And I guess the question is what value that is to the industry at large. I mean, is industry willing to actually help to fund the work that it's going to take to get this there?
Right now, as I think I mentioned, Taylor's is one of the largest shellfish farming companies in North America, and we have a whole component to our company which is based on research, and so we end up putting actually quite a lot of our own dollars in kind, and sometimes actually money and staff into research. So yeah, in the past we have funded stuff. In each case we have to look at it. As far as other than just yield, husbandry is fairly basic with shellfish as far as different breeds of the same species, and we suffer significant losses, sometimes definitely in the hundreds of thousands of dollars, sometimes in the millions of dollars, with die-offs or reduced growth. And understanding sometimes quite a few of those die-offs we don't fully understand what happened. There's one that's been ongoing. There's a problem here in France called summer die-off in oysters, and it's pretty general on the west coast, and it's a big problem in parts of France, and it's actually very poorly understood why that happens. It seems to happen with the fastest growing oysters. With mussels, we had the Trombrose Milscala Fugialis here. It's probably a little cold for it here, but then, as Helen mentioned, understanding Edulus and Gallo, some Patrick species. Some people call them variants of the same species, and some people call them separate species. But so understanding how to get the best animal to grow here could really help make the industry much more viable. So yeah, thank you. If I answered the question right.
And like maybe if I could add from like a technology point of view, we know so little really about the function of these organisms, and so you start initially at a broad scale microarray experiment, and then you try and narrow it down using maybe real time. And at the moment we're in discussions about developing a proteometric kit, so that we can actually then have something that somebody in the field can use. So it's moving the technology away from kind of people like me and down more into people like Gordon to use.